Professor Adeola Kola-Mustapha was born on 20th August 1974 to Alhaji Adekunle and Alhaja Adeola Lasisi in Lagos State, an indigene of Osogbo along the local government area of Osun State. She attended Tuwashe Nursery and Primary School and Adeni Jones Primary School in Ikeja, Lagos, for her primary education, and Federal Government Girls College, Ilorin, Imo State, for her secondary school education. Professor Kola-Mustapha gained admission to study pharmacology at the University of Lagos in 1992 and graduated with Bachelor of Science Pharmacology first class degree. She furthered her studies with a Bachelor of Pharmacy B.Pharm and a Master of Science in Pharmaceutics and Pharmaceutical Technology, graduating with distinction, all from the University of Lagos. She obtained a PhD in Pharmaceutics and Pharmaceutical Technology from the Montfort University, Leicester, United Kingdom in 2014. She holds a postgraduate diploma in education from the University of Ilorin in 2017 and PharmD degree from the University of Benin in 2019. Professor Kola-Mustapha joined the services of the University of Ilorin in the Department of Pharmaceutics and Industrial Pharmacy as a Lecturer 1 in 2015 and rose through the ranks to become a Professor of Pharmaceutics and Industrial Pharmacy in 2024. Before joining the service of the University of Ilorin, she worked as an intern pharmacist at the 68th Nigerian Army Reference Hospital, Yaba, Lagos. While pursuing her PhD at the Montfort University, she worked as a part-time lecturer and technical demonstrator at the Leicester School of Pharmacy between 2010 and 2015. She also worked briefly with Leeds Pharmacy, Oxford, United Kingdom, as a research and health service development manager in 2010. She has also served as a consultant associate professor to the College of Pharmacy at Al Faiza University, Riyadh, Saudi Arabia. Professor Kola-Mustapha successfully supervised several undergraduate and postgraduate students. She is a member of many learned and professional bodies, and these include Pharmaceutical Society of Nigeria, PSN, International Pharmaceutical Federation, American Association of Pharmaceutical Scientists, United States, Canadian Society of Pharmaceutical Scientists, the Academy of Pharmaceutical Science, Association of Pharmaceutical Scientists, United Kingdom, among many others. She has served in various capacities within and outside the university, and these include acting head, Department of Pharmaceutics and Industrial Pharmacy from 2019 to 2022, Council member, Medical Science Representative, Science Association of Nigeria 2018 to date, Coordinator, Department of Pharmaceutical Microbiology and Biotechnology 2016 to 2017, Member, Postgraduate School Committee 2021, to mention but a few. Professor Kola-Mustapha is a recipient of several academic awards, which includes Fellowship Award by the Science Association of Nigeria at the 57th Annual Conference by University Kano 2025, Role Model Award by Pharmaceutical Society of Nigeria, Kwara State 2023, First Prize Award at the FISB Symposium on COVID-19 Research Italy 2020, Merit Award winner from the Pharmaceutical Society of Nigeria, Kwara State 2020. Professor Kola-Mustapha has over 50 publications in reputable home-based national and international journals, edited conference produced as chapters in books. She is a scholar of exceptional merit, a researcher of international repute, and a dedicated teacher. She has made indelible contributions to the field of pharmaceutical sciences. Her groundbreaking research in drug delivery systems and herbal therapeutics has not only advanced scientific knowledge, but has also provided practical solutions to critical health challenges. Professor Adeola Kola-Mustapha is happily married to to Savio Olukolade Kasim, and the union is blessed with children. Let me invite Professor Adeola to Kola-Mustapha to deliver the 250 inaugural lecture for our university. Congratulations.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم قال رب اشرح لي صدري ويسر لي أمري. The Vice Chancellor, the Deputy Vice Chancellor Academic, the Deputy Vice Chancellor Management Services, the Deputy Vice Chancellor Research, Technology and Innovation, the Registrar, the Bursar, the University Librarian, the Provost, College of Health Sciences, Dean of the Faculty of Pharmaceutical Sciences, Dean of other faculties, Postgraduate School and Student Affairs, professors and members of the Senate, directors of various units, head of Department of Pharmaceutics and Industrial Pharmacy, academic and non-academic staff of the university, my lord spiritual and temporal, distinguished invited guests, gentlemen of the press, students of the Faculty of Pharmaceutical Sciences and other students here present, great students of the University of Ilorin, distinguished ladies and gentlemen. All thanks to the Almighty Allah for the opportunity He gave me to be here before you all today. With profound gratitude, I'm honored to present the 285th inaugural lecture of the University of Ilorin, the first lecture for the Faculty of Pharmaceutical Sciences titled Pharmacokinetics Essential Tool in Drug Delivery Therapy was delivered by the esteemed Professor Mujiti Bakari Adeola on the 7th of November 2013. The second lecture, titled Drug Formulation as Essential Construct for Safe Delivery, was presented by Professor Ilorin on the 14th of March 2024. Today, it gives me great pleasure to deliver the third lecture precisely from the Department of Pharmaceutics and Industrial Pharmacy on the topic, Rooted in Nature, Refined by Science, a New Horizon in Advanced Drug Delivery. With this title, I plan to synthesize useful findings from more than a decade of research since my PhD into a lecture that I hope will enlighten the listeners today on the vital role of pharmacists in advancing healthcare for all. My winding sojourn today began with my first undergraduate degree, BSc in pharmacology from the University of Lagos. Driven by my passion for pharmacy, I returned to study pharmacy at the same university where I obtained my second undergraduate degree, B.Pharm. After my one-year internship, I enrolled for the master program and obtained an MSc degree in pharmaceutics and pharmaceutical technology at Unilag. My PhD journey began at the University of Lagos, but later transitioned to the Montfort University in Leicester, UK, where I worked as a technical demonstrator and a part-time lecturer. In addition to my rigorous research, inspired by my interest in impacting knowledge at this stage and a commitment to contributing to Nigeria's educational sector, I was privileged to take up a lecturing commitment to contributing to Nigeria's education. I was privileged to take up a lecturing position at this prestigious university, University of Ilorin, the Better by Far University, where I have served diligently since 2015. It would be impossible for me not to appreciate my wonderful family, friends, mentors, and colleagues, too many to count, who have made this journey possible. Mr. Vice Chancellor, sir, this lecture is an interwoven piece of my research and the process of pharmaceutical innovation in improving patient outcomes towards saving lives. A pharmaceutical scientist specializes in the research and discovery of novel drugs and drug formulations for improved therapeutic outcomes. In this vast section of discipline, I have devoted myself to the development of pharmaceutical formulations and enhancing their delivery in enabling optimum activity of active drug ingredients. Drug formulation, a critical component of pharmaceutical development, is the combination of an active ingredient and other non-active ingredients into a drug that meets acceptable standards of quality, safety, and efficacy. Mr. Vice Chancellor, perhaps it would be useful to use the simple analogy of jollof rice in explaining this concept. The rice itself is the active pharmaceutical ingredient, but since it cannot be served bare, you add condiments that make it palatable to the consumer, Nigerian or Ghanaian. These condiments are the excipients, the process of boiling and frying that take place before the food is finally served correspond to the different stages of pharmaceutical formulation. Away from food and back to drugs. We commonly see a classification that divides medicine as either natural or synthetic. Natural medicines are defined as therapeutic products obtained from various natural sources, including plants, animals, and minerals that are useful for managing or treating the illnesses. Vice Chancellor, sir, permit me to clarify to the audience that all herbal medicines are natural drugs, but not all natural drugs are herbal remedies. Herbal remedies are plant-derived and only one of the various sources of natural medicines. On the other hand, synthetic medicines are artificially produced or modified from natural principles. They are also useful in treating or managing diseases. While these definitions make it clear that a drug is either here or there, it is not always the case, and in many cases, a synthetic drug may still contain natural components in its excipients for formulation and improved delivery. The mention of natural drug or traditional medicine often evokes an imagery of agbo or concoctions in suspicious-looking dark-colored preparations in transparent bottles. And when I say synthetic, what may come to mind is the image of a well-labeled solid or liquid drug in a neatly arranged pharmacy. I want to begin by clearing away these prevalent assumptions and help to draw a bridge between the two stark classifications. Clearing the myths. Natural is dirty, synthetic is clean. All drugs should be clean and prepared in an acceptable form that meets standards for quality and safety. The unclean conditions for sale of many natural drugs are only reflective of the gaps in regulation and control. Another myth, all natural drugs are safe. A natural product may be beneficial, but if used beyond the stipulated limit, it can also harm the patient. Also, a poorly prepared natural product may contain harmful toxins in addition to the beneficial plant extracts. Therefore, a natural product should be used with the same caution as a synthetic medicinal product. Another myth, natural drugs and synthetic medicines cannot coexist. Far from reality, it is almost impossible for natural and synthetic medicines not to coexist, but it is the degree of integration that may matter more to healthcare advancement and national empowerment. We have seen relevant examples of countries, China and India, utilizing both forms of medicine in mainstream practice. Indeed, the side-by-side adoption of natural and synthetic medicines is beneficial for national health. However, let us think of a deeper level of integration. Mr. Vice Chancellor, can we go beyond the side-by-side implementation and move into a wholesome combination where the natural product is indistinguishable from the synthetic product and rather constitutes a vital portion of it or an entirely new one? Certainly we can, and here's where the lecture begins. The inherent gaps in synthetic medicines. Literature is replete with reports of formulation complexity in addressing inherent gaps of synthetic products. The determination of the activity of the compound is not enough to guarantee its use in patients. In some cases, these active pharmaceutical ingredients API may possess a bitter taste or poor solubility that creates a challenge in formulation. It is up to researchers in the pharmaceutical science space to determine the most efficient formulation that ensures stability and appropriate delivery in human systems. This formed the basis of my PhD thesis and the early year of my research experience. The solubility of drugs play a key role in its pharmacokinetics. Mr. Vice Chancellor, there are numerous poorly water-soluble drugs. Permit me to use ibuprofen as a prototype drug onwards. Ibuprofen is a common non-steroidal anti-inflammatory drug with analgesic, anti-inflammatory, and antipyretic properties. It is commonly prescribed as a pain relief in pharmacies across the country. What you may not know is that ibuprofen is practically insoluble in water. It belongs to the BCS class 2, high permeability and low solubility, and this makes it difficult for it to be prepared as a solution. Due to its high coercive nature, it floats poorly and does not exhibit good compressibility, making it difficult to be compressed as tablets. Adeola, Kola-Mustapha et al. 2014. Typically, the granulation method is used in improving the probability of powders when preparing tablets. This method involves the aggregation of small particles into larger spherical particles, where each original particle is still identifiable. This method minimizes dustiness and material loss, but does not solve the problem faced in manufacturing and those that emerge unpredictably in ibuprofen's product life cycle. Adeola and Kola-Mustapha 2015. As a well-conceived response to these difficulties in utilizing ibuprofen, Adeola, Kola-Mustapha and Ruparelia in 2014 attempted to develop a polymer drug conjugate to enhance its solubility property. Ibuprofen, which is an amplifier, was used, eliminating the need for any organic solvents. The complex ibuprofen melt solution and diethylamino ethyl dextran DDEX to prepare stable amorphous ibuprofen polyelectrolyte crystal granule conjugates, which is a crystal, or using the melt in situ granulation crystallization with temperature quenching technique, and this is shown in figure 1. In the same manner, DDEX conjugation increased the in vitro ibuprofen release rate, and is shown in figure 2. Artificial neural networks showed an excellent correlation between predicted and experimental data in solubility and dissolution. In addition, particle rearrangement was improved in the crystals. Overall, the DDEX conjugates of ibuprofen exhibited a better pre-compression profile than the pure ibuprofen. Mr. Vice Chancellor, what started with the problem of poor water solubility of ibuprofen led to the development of a polymer drug conjugate, specifically with cationic diethylamino ethyl dextran. We did this without the need for toxic organic solvents commonly used in the mainstream production of pure ibuprofen. Furthermore, Adeola and Kola-Mustapha 2015 characterized the ibuprofen DDEX conjugate granules, and the surface morphology characterization results are shown in figure 3 showed that the ibuprofen DDEX conjugates exhibited plate-like agglomerates that were well-defined and non-symmetrical in shape, unlike the pure ibuprofen with a regular surface. Taking a step further, our next objective was to optimize ibuprofen formulation by another approach, distinct with the earlier described drug granulation method in ibuprofen DDEX conjugates as presented in table 1. Here, we developed ibuprofen dextran conjugates by surfactant solubilization method. With this, we observed that the organic conjugates exhibited surface activity and high probability of reducing the surface free energy, which could enable increased solubility. Additionally, we were able to achieve high loading capacity and reduce particle size of ibuprofen, Kola-Mustapha and Adeola 2015. From this study, we were able to conclude that the ibuprofen dextran and the conjugates possess preferred properties in drug release when compared with pure ibuprofen, as shown in figure 4. Polymeric film proteins offer another option in drug development, especially in sustained drug release and masking unpleasant drug taste. In 2016, Kola-Mustapha et al. Sought to improve the integrity of aqueous polymeric films via the use of polyelectrolyte complex. We used gelan, which is a natural polysaccharide with negative charge for the formulation. Results from this research effort showed that drug loading capacity and conjugation efficiency increased with increasing DDEX concentration. On introduction of this DDEX in conjugation, the surface began to exhibit opaque and rough features, and the opacity increased with additional DDEX. Some of the nanoconjugates also grouped together to form fluoride aggregates, as shown in figure 5, which increased additional levels of DDEX, either due to the increase in adhesiveness or non-electrostatic interaction. Summarily, we were able to develop optimized ibuprofen conjugate polymeric films with increased loading capacity and efficiency in dissolution. Now, with while polymeric conjugates are useful in enhancing the delivery of polysoluble drugs, synergism with hydrotropic complexation, this resulted in a marked increase in drug release, as shown by Kola-Mustapha and Adeola in 2018. And the figure of the FTIR is shown in figure 6. Adeola and Amitej Kola-Mustapha in 2015 used another polysaccharide. So, Vice Chancellor, sir, what if we could try a different polysaccharide? And then we went on to use chitosan by developing an amorphous nanoparticle complex with ibuprofen. Compared to pure ibuprofen, the formed nanoplexes exhibited faster and extended release profile, with the major means of drug release being diffusion amidst the occurrence of anomalous transport and super-K2 transport. Then, Kola-Mustapha and Adeola also developed and characterized a novel polyelectrolyte complex hydrogel from gelan gum and dextran. Again, the complementary utilization of synthetic and natural components in advancing drug delivery saved the day. Now, piroxicam, which is another NSAID with poor solubility properties in water. With this drug, Tela, Inaugural, and Kola-Mustapha and others in 2020 tested another mechanism in optimizing delivery in the body using a novel class of viable hybrid materials called the metal-organic frameworks, the MOFs. In our study, Tela, Inaugural, Kola-Mustapha successfully loaded a piroxicam on the zinc and copper MOFs, and they were further tested for solubility, drug release, and dissolution properties by Kola-Mustapha, Ayuba et al. In 2020. and the results showed that zinc mobs had the highest release for piroxicam. Vice Chancellor, sir, synthetic drugs are inherently imperfect and may often require special formulations processes in optimizing delivery. Some of these optimization processes involve the use of natural products such as gelan and chitosan, as seen with some of our work with ibuprofen. These drug delivery research efforts to advance the application of existing drug molecules is a continuous quest for me. The objective is to have some of the mentioned techniques integrated into the Nigerian local pharmaceutical scene in the nearest future. Usefulness of natural products. Mr. Vice Chancellor, nature has blessed humans with a countless numbers of plants with effective phytochemicals for managing various illnesses. Sometimes, one single plant may even be useful in 2 or more diseases. The bark may be used for disease A, the leaves for disease B, and the root for disease C. In parallel with working on synthetic medicines, I've also peered into the bioactivity of natural plants, formulation, and their potential benefits for patients. In Nigeria, there is an increasing interest in natural medicines, especially herbal formulations across all age groups. Ghazali, Bello, and Kola-Mustapha in 2019 carried out a study to determine the use of herbs among our patients at the University of Ilorin Teaching Hospital. And we realized that all patients knew about herbal medicines. About 68% used them, 25% were currently taking herbs. Over half of them did not follow specific dosing. Many combined herbs with additives or synthetic drugs. About 67% reported that the illness were resolved, 13% said no effect, and 20% said it gave temporary relief. This study highlights widespread herbal medicine use, often alongside conventional treatments, underscoring the need for standardized dosing, safety evaluation, and integration strategies in Nigerian healthcare. Kola-Mustapha et al. In 2023 carried out a study in Kwara State among traditional medicine practitioners to obtain a list of herbal therapies used in managing sickle cell disease in the state. Working with 37 traditional medicine practitioners as study participants, we gathered the following list of plants as shown in Table 2, identified as sickle cell disease therapies. This highlights how nature's vast local botanical resources can be identified and channeled to address sickle cell disease. And this remains a significant public health challenge in Nigeria. The following herbals were studied by our team: Kambisi, Bakari Adeola, Kola-Mustapha et al. In 2017, Hibiscus sabativa, popularly known as Zobo, Moringa oleifera, Ophimum grattissimum, known as Ephedrine, and Telferia occidentalis, known as Ugu. And we found that the tea occidentalis exhibited about 72% DDPH antioxidant activity and 58% adenosine peroxide scavenging potential. And this was greater than Moringa, Ophimum grattissimum, and Hibiscus sabativa. Integration in drug development and formulation. Mr. Vice Chancellor, I began this lecture with an illustration of an imagery that is prevalent in our minds when we talk about natural medicines used side by side with synthetic medicines. This common imagery sometimes limits the use of natural medicines, even when they are efficacious for an ailment. I envision a future where synthetic and natural medicines are indistinguishable from each other, both appealing to the user with efficacy and safety for use in patients. Such a future would entail the formulation of natural products as finished pharmaceutical products where herbal constituents are prepared in synthetic dosage forms such as emulsions, capsules, and tablets, just as it is done in advanced parts of the world. In the most recent part of my research career, I've focused on expanding novel technologies in synthetic formulations to optimize natural medicines with proven efficacy. About gels. Entandrophragma italy is a flowering plant commonly known as Ijeboy Yoruba. It is a part of the Malaceae family and is widely prevalent in West Africa. Kola-Mustapha Ibrahim et al. In 2023 sought to develop an emulgel of this plant as a topical dosage form for the management of inflammatory conditions. When tested on a chronic inflammation and animal model as shown in Figure 7, the treatment resulted in a dose-dependent effect. The emulsions were comparable with the standard diclofenac emulsion in efficacy. This study provided a useful rationale for the integration of natural medicines in synthetic formulation methods. Nevertheless, essential oils are aromatic compounds obtained from various parts of plants and are usually very volatile. Melaleuca telefonia tea tree and Subopogon flexosus lemongrass are common essential oils with proven activity against various microorganisms. Kola-Mustapha et al. In 2023 sought to combine both essential oils in developing a synergistic formulation for the treatment of vulvovaginal candidiasis. When combined to form an emulsion preparation, we observed a synergistic effect resulting in better activity against implicated fungi. When applied against Candida albicans in this study, the emulsion diffused to release the essential oil components as shown in figure 8. The formulation was also stable and showed satisfactory characteristics for vaginal use. In another research, Kola-Mustapha, Abdurrahman, and Ishola in 2023 sought to develop Actium lapa root extract-based emulsions for the potential management of chronic inflammatory disorders. And the Actium lapa is utilized in multiple continents for the treatment of chronic inflammatory conditions. It is reported to be beneficial in managing pain from rheumatoid arthritis, but it is linked to unfavorable side effects after oral use. So, Kola-Mustapha, Abdurrahman and Shola developed an emulsion system with internal extracts of the roots of Actium lapa for topical use. When tested for activity, the herbal emulsion produced significant results in anti-inflammatory activity. We also achieved good spreading coefficient and desirable viscosity for this dosage form. Mr. Vice Chancellor, there's a high prevalence of rheumatoid arthritis in our society, especially among the elderly, and we must consider natural remedies in pain and inflammation management. This motivated my colleagues and I to consider more plants with viable potentials for formulation into an emulsion for easy use and application by patients. Terminalia macrotera, known as Ponderi, is traditionally used for various elements in West Africa. Ibrahim, 2005. Kola-Mustapha et al. 2023 conducted the anti-inflammatory activity of the extract, formulated into emulsions, and tested their physical characteristics as shown in table 4. The optimized emulsion showed anti-inflammatory activity comparable to diclofenac, indicating its potential as a treatment for rheumatoid arthritis. Mr. Vice Chancellor, pain is a global phenomenon. From headaches to the bone strains, the extent of discomfort suffered in pain demands the production of better analgesic formulations. Based on the reality, we developed another plant-based dosage form for its analgesic and anti-inflammatory properties. Kola-Mustapha, Johanna et al. 2020. Chasmantera dependence and Chenopodium apophysoides are plants reported to be useful in treating pain and are being used by traditional bone setters. Leveraging on these prospects, an efficacious herbal gel was developed with desirable physical and pharmacological properties as shown in figure 9. Kola-Mustapha and Khalid Salako, 2021, developed another emulsion for the management of rheumatoid arthritis using Kola milani, Obiedin in Yoruba or Oto in Igbo. The formulated emulsion, as shown in table 5, exhibited a dose-dependent activity comparable to standard diclofenac emulsion with good pH and extradability values and a notable ease of spread. If all these plants are not familiar, then you should know Zingiber officinale, known as ginger, and Oxymum basilicum, known as Ilorin. With the essential oils of these two plants, we developed an oral gel for the management of oral candidiasis. On testing against Candida albicans, Kola-Mustapha, Adeola, 2020, observed that the diameter of the zone of inhibition went from 23 to 32 millimeters for Zingiber officinale and 22 to 26 millimeters for Oxymum basilicum. When combined, the highest activity was seen at a ratio of 25 to 75 for ginger and basil essential oils. After formulation, the emanating herbal gel was effective in inhibiting the growth of Candida and has been recommended for extensive tests and subsequent clinical trials. Yes. Now to herbal creams. Sibapitandra, known as Ogungwa Araba, and Lania Kasingi, 2D, are traditional Nigerian plants used for various ailments like diabetes, asthma, ulcer, and hemorrhoids. Combining both extras, Aremu, Kola-Mustapha, and Ayotunde, 2017, developed a cream using a screen base and evaluated its properties and efficacy. It exhibited a potential for treating skin conditions caused by Trichophyton species. For herbal suspensions, Mr. Vice Chancellor, our natural flora remains an endless source of medicines in managing our ailments. Diarrhea is the leading cause of death among children and with adults in Nigeria. Kola-Mustapha et al. 2019 identified a plant with reported activity in managing diarrhea, and that's Pacotina nagricense, a plant native to various parts of Africa and traditionally used to treat diarrhea. We aim to develop a standardized oral suspension for the consumption of this medicinal plant to aid dosing and guarantee safety after use. The formulated super suspension with excipients which were natural and synthetic minimized sedimentation and demonstrated effective stool control in animal tests as shown in Table 6, offering a promising natural remedy for diarrhea management. Herbal suppositories. Vice Chancellor, sir, would we be pleased to know that herbal products can be formulated into other dosage forms as well, depending on the intended means of administration. One example is suppository. Kola-Mustapha, Bamigore and et al. In 2021 extracted black seed oil, characterized and determined its antifungal activity. The resulting suppositories demonstrated effective antifungal activity, highlighting their potential as alternative herbal treatments, as shown in Figure 10. Novel drug delivery. Central to the theme of natural and synthetic medicine integration is the design and development of novel drug delivery systems that enable the co-utilization of both medicines either for synergistic impact or for supporting the activity and appeal of either to the consumer. Evidence in this lecture is the recurrent thread of nanomedicine, whether it is the mention of nanoconjugate or the mention of nanomatrix films. Since the portability of drugs is a common concern in drug development, nanomedicine offers a great benefit in solving this problem through nanoconjugates. An amorphous polymer drug nanoconjugate is a hybrid of an active drug physically or chemically linked to a polymer from which it can be cleaved for release in the biological systems. Adeola, Kola-Mustapha et al. 2016. Nanoconjugates have been successful in reaching the market since the 1990s and can be developed through the different methods. Despite the level of progress in research seen so far, as shown in Figure 11, there's still room for the use of this drug delivery system across various ailments. Another nano approach of interest is the use of nanocellulose and hydrophilic metal oxide composites. Olatunji Saliu, Kola-Mustapha et al. 2017 studied the delivery of tramadol hydrochloride by regenerated cellulose nanofiber titanium oxide zinc oxide composites. Combined with tramadol, we aim to develop a soluble hydrogel matrix for the sustained release of the active to enhance compliance and reduce habit forming. Drug release kinetic studies showed that the composite with the least portion of zinc oxide and titanium oxide had the highest release percentage of tramadol. As the amount increased, the drug release slowed down and was sustained. Nano delivery is important for its ability to enable the delivery of drugs to specific bite sites in the body. Conventional medications are often limited in their ability to cross the blood-brain barrier, and this is in most cases beneficial to prevent side effects on the brain. However, in the treatment of brain conditions, the drugs must be able to get to the brain. Hence, there's a need for nanodrug delivery systems that can bypass the brain's barrier in the treatment of diseases such as Parkinson's, Alzheimer's, and others. Kola-Mustapha, Adedeji et al. 2023. Nanotechnology has also been shown to be useful in enhancing the viability of lactobacillus species, a common probiotic for vaginal candidiasis. Ilorin, Ogundemure, Kola-Mustapha et al. 2024. Via the electrospun nanofiber-based delivery system, we were able to develop lactobacillus-related nanofibers that did not cause any irritation to animal subjects and preserved the viability of the bacteria. Computational drug analysis. Vice Chancellor, sir, the world is digital and research processes are quickly evolving with novel computational tools that improve output and offer useful insights. In the past decades, it was impossible to know if a drug would work for a particular disease without testing it on an animal model. Nowadays, there's software that can predict the activity of a compound in vivo, and it supports safety and toxicity effects. And you can do all this from the comfort of your office without entering the laboratory. However, additional laboratory tests are useful to validate computational results and enhance research robustness. Computational drug analysis leverages the digital tools to predict a compound's activity, safety, and toxicity without immediate laboratory testing, enhancing research efficiency. Using this approach, Elsewedi Asheri Kola-Mustapha et al. 2024 designed structural derivatives of eugenol, a natural phenolic from cinnamon oil, which improved antibacterial activity against DNA gyrase, as shown in Figure 12. This study highlights the value of computational methods in drug design and potential to develop novel therapeutics from natural products. In another study, Kola-Mustapha in 2023 employed similar tools in the de novo design of pimarine diterpenol compounds as potential therapies for the treatment of acne vulgaris. By modifying the structure of pimarine compounds, a new compound was designed with better affinity for the drug rather than drug target than salicylene, which is the standard. This study showed that computational drug design and discovery offer a useful way to validate the activity of natural and nature-derived compounds and optimize the associated drug development process. Contributions to the university. In my admission to this position, I consider service in mentorship and student development as my greatest contribution to the university. My love for academia is motivated by the desire to not only train students intellectually, but also morally and other phases of extracurricular development. I have served in various capacities as a teacher, researcher, and collaborator on various projects. My service to the academic, non-academic, and global research community at large is not exempt from this. From initially starting out with teaching courses on pharmaceutics to undergraduate students to now supervising postgraduate students, this maxim is clear. To whom much is given, much is expected. Some of the positions I have served at the peak, I am the member of this university senate, University of Ilorin, and at the start, I was an examination officer, Department of Pharmaceutics and Industrial Family, and then research manager, Faculty of Pharmaceutical Sciences. Conclusion. Mr. Vice Chancellor, sir, as I stand before you today, I'm in awe of my growth and the development of the academic community at this prestigious university. In about a decade of service to this institution, I have risen through the ranks to become a professor of pharmaceutics and industrial pharmacy. Being the first female professor from my department, it gladdens me to inspire young women in the sciences. For emphasis, natural and synthetic medicines are complementary. They have always been. By formulating natural medicines in synthetically appearing formulations such as emulsions and suppositories, we define the user's perception of traditional medicine and enhance its use in the population. By improving synthetic medicines with naturally occurring excipients, we strengthen its effects and alleviate drawbacks in its production and use. By combining both for activity, we achieve a synergism in effect that exceeds the sum of its parts. Lastly, the achievement of this can only be possible with the embrace of novel trends and developments in pharmaceutical formulation and development. Computational drug analysis with bioinformatic tools is here to stay and is now a gold standard in many countries. In the end, the goal is to use what we have, synthetic medicines, in conjunction with what we have been given, natural medicines, to produce what we direly need, healthcare advancement. Our research niche is being achieved with materials that are either primarily or secondarily derived, rooted in nature, refining them with the aid of pharmaceutical technologies, including bioinformatics, and making them better drugs and excipients for advanced drug delivery. Recommendations. Mr. Vice Chancellor, ladies and gentlemen, here are my recommendations based on this lecture. 1-in-1 and not side by side. The goal of integration is not to place natural and synthetic medicines side by side in a pharmacy, but that they are co-developed until one is indistinguishable from the other. Indigenous pharmaceutical formulation practices should aspire to equal standards of quality, appeal, and activity for natural and synthetic medicines, and continuously attempt the co-formulation of both for the better health of Nigerian patients. No medicine is perfect. To the audience, every drug is a poison and should be used with caution, whether natural or synthetic. To my fellow scientists, let us bring to the fore prevalent issues in medicinal development and use in patients. Scientists should continuously strive to identify active plant components, determine safety, and standardize those, and further improve drug formulation for industrial efficiency and patient benefit. More than this, there is more to pharmaceutics than mortar and pestle. The modern world introduces students to cutting-edge technologies for formulation development, setting the pace in the next wave of industrial revolution. From computational hardware and software for producing drug activity to the use of artificial intelligence in optimizing the drug development pathway, there is a critical gap in the Nigerian healthcare system. Public and private partnerships are encouraged to accelerate access to these technologies in pharmaceutical training. Using what we have. It is not enough to test these formulations in the laboratory. It is more important that we commercialize for use. Bridging town and gown is a common term to describe research translation to societal use. However, I believe that we can achieve even more significant breakthroughs where there is no longer a need for a bridge between the laboratory and the industry. It will be of great benefit to the university to consider the development of in-house production capacity for the commercialization of novel medicinal formulations. I am sure that the Nigerian populace is hungry for made in Nigeria and will not hesitate in patronage of the university's products. Acknowledgements. Oh Allah, you alone deserve to be praised in granting this elevation, which is the hallmark and pinnacle of academic profession. I thank you for your infinite mercies. Alhamdulillah Rabbil Alameen. Thank you, my Vice Chancellor, sir, Professor Abdulwahab Alashuko Egbewole, for granting and supporting the delivery of my inaugural lecture. Sincere gratitude to the Deputy Vice Chancellors, Registrar, Bursar, and University Librarian. May Allah continue to have mercy on late Professor Abdulkarim Agunu, the Dean of Faculty of Pharmaceutical Sciences till 2017. I gained a friend and a sister in his wife, Mrs. Umma Abdulkarim, and the children, Zainab and Ahmed. May Allah bless you all. I acknowledge all the past deans of the faculty, Professor P.F. Oluyola, Professor Ajara Ibrahim, Professor Rashida Tayoni, Professor Ayo Aremu, Dr. Muhammad Amali, Dr. Sahad Abdullahi, and my current dean, the Dean of the Faculty of Pharmaceutical Sciences, the Dean of the inaugural lecture of today, Professor Abigani Giwa. Thank you most sincerely to my sub-dean, Dr. Owai Enayewu. Thank you for all you do. To Dr. Shitu, my current HOD, thank you for the selfless service in leading the department. A hearty thanks to all my amazing colleagues in the faculty here listed. Not forgetting my laboratory technologies colleagues, led by Mr. Utayo, and other department technologists who made my bench work, teaching, laboratory session, exam administration seamless and successful. Accept my sincerest appreciation. Your efforts are very much appreciated. The past and current non-academic staff colleagues, ably led by the faculty officer, Alhaji Abdulwahab Bello, and all who served in different capacities to support me in the Department of Pharmaceutics and Industrial Pharmacy, the faculty, and Ilorin at large. Thank you. Thank you, Professor Mojibakari Adeola and the Akri Research Group. I appreciate working with senior colleagues here listed. Thank you, Professor Yusuf Adedayo Abdulkarim, former DVC Management Services, for the inspiration of my PGD thesis and mentorship. My genuine thanks to the members of the Ilorin compatriots, our past and current conveners, esteemed members of the Ilorin Women Association. Thank you, Professor Alhaja Oba Abdurrahim. I thank the Chief Imam of Ilorin, the Muslim community, and members of Muslim Ladies Circle for their immense support. Thank you, my brothers and sisters here mentioned. There's a long list, but I thank everyone. I each thank you to my immediate past head of the Library and Publications Committee and the current for their immense support and coordination of this inaugural lecture. Thank you to my editorial team, Korede Adedeji, Yusuf Ghazali, Sheyi Egbawande, and Miriam Isiaq. Thank you for your time and support as always. I sincerely thank my senior colleagues, sabbatical staff, and external examiners who have served as mentors and are here listed. It's a long list from Professor Musa Adebayo to Professor Ayinde. My warmest thanks to my colleagues who have served as peer mentors, both support system and research collaborators, Professor Petra Namani, Mr. Uche George, Dr. S. Katibi, Dr. Ebesi Abo, Dr. Ike Bello.
Thank you especially Professor Chidebin Bruce Babalola, the CDDDP, UI, and USPQM team, Dr. Kosula and others, and USP facilitators from Mopa Suga, from Tony Longo and Dr. Jonathan Okuru. Thank you. Thank you to Professor Mojisola Adeyeye, the current NAFDAC DG for the research collaboration opportunities provided. I appreciate my supervisees, undergraduate early said, from Amida Ayotunde, Yusuf Gazali, Aisha, to everyone listed here, and my graduate students, Oluwase Isali, Ola Olatunji, and Miriam Aruna Baker. I thank Professor G. Olatunji and Professor S. Oguntoye for the opportunity to co-supervise MSc and PhD candidates for the Faculty of Physical Sciences. Thank you to all undergraduate and graduate students whom I have taught over the years, who gave me the opportunity to improve my teaching skills and enjoy what I love to do best. I'm looking out for you all, my pharmacocos. Your success is indeed my success. I pray you all surpass my achievements. Amen. Thank you, Pansites, for initiating the annual Professor Kola-Mustapha Medical Outreach Program. My mentees, their spouses, and their families early said, it's a long list. Thank you. I say a very big thank you to you all for immeasurable support. To Yusuf Babatunde, Korede Adedeji, Abdulmumin Ibrahim, and Melody Okoroke, thank you for challenging me with reviewing your pitches and grant applications. To my esteemed undergraduates and graduate alumni, to the faculty, the YPGs and PSM members, I'm indeed proud of your achievements thus far. My heartfelt gratitude goes to everyone who supported this journey. Your efforts have been truly impactful. To my parents, Alhaji Adekunle and Alhaji Adeola Lasisi, thank you for your unwavering support and guidance. It is an honor to have been blessed by you. You stepped into taking care of my children when the inevitable happened. Your selfless care is invaluable. Indeed, Iyaniwura Iyebiye Babani Jigi. My Lord, be merciful to them as they raised me when I was small. Quran 17:24. Amen. To my siblings, their spouses, Balaji and Taiwo, Folagbade and Adeola, Ismail Aziz, Adepeju and Tajuddin, and children, Farida, Faiz, Adam, Ali, Faz, Toyib, and Chakia. I'm grateful to be part of such a loving and supporting tribe. Thank you for cheering me on, as always. I love you all profoundly. To our in-laws, Adenekos, Ogushola Iroruns, Serikis, Shomades, and Kasims, Ajoshewa Ogubaje. I appreciate from Elijah Rizikans, the matriarch of Kasim family, to Ajia Tony Idris, my husband's siblings, and Sorias Femi Kasim, FNIS. Thank you so much. To my uncles and aunties and their children, the Aziz Lasisi, Ajibade, Gambari, Badamosi, Lawal, Adeyemi, Shitu, and Akobin families. You are all indeed exceptional. To my cousins, the Aziz, Adekus, Ajibades, Zubaros, Idorus, and Salamis, thank you for the fun times and loving relationship. May Allah continue to have mercy on Alhaji Junai, Olatunde Adeniji of Tabade Pharmaceutical Chemist Limited, for his fatherly role and priceless legacy of mentoring me alongside other pharmacists. I thank Alhaji Ayabadeji for our wise counsels and support always. My deepest appreciation also goes to my teachers during my formative years at Tumashe Nursery and Primary School in Ikeja, Adini Jones Primary School, Masjid Akhari Madrasa Seriki Awe, Ikeja, Lagos, and Federal Government Girls College of Imo State. Thank you to Professor Abdulfatah Abiola Madawadeje and my teachers at the Department of Pharmacology and Toxicology, College of Medicine, Unilag, where I obtained my BSc Pharmacology degree. Thank you to Professor H.A.B. Increase Coker, who ushered me into the School of Pharmacy, Unilag, via direct entry. My supervisor for BPharm, MSc initial years of my PhD in pharmaceutics, Professor Ndui Ifudu. I deeply appreciate your fatherly role and support. My sincere gratitude to all my lecturers at the School of Pharmacy, College of Medicine, University of Lagos. To my PhD body and prayer mentor, Dr. Margaret Ilomwaya, thank you. I also appreciate my esteemed collaborators from my alma mater, Dr. Ismail Ishola, Dr. Adejimon, Dr. Ibrahim Oseni, Professor Milabu, and Professor Oyagba. A very big thank you to my Egbon from his residency days at Lut, Professor Biodun Biman Alabi, the provost of the College of Health Sciences in Ilorin. I remain truly grateful to my main PhD supervisor, Dr. Amos Olusegun Abiri. I'm sure he's listening to me online, for the awesome mentorship and guide through research design, writing, and publishing through my PhD sojourn at the Montfort University, Leicester, UK. Thank you, sir, for always checking on my progress. Thank you for the supportive men of my core supervisors, Professor John Taylor online, Professor Volkia Swindline, Professor Von Perry of the Athens University, Birmingham, and Professor Barbara Conway of the University of Huddersfield. Thank you also to my To my mentees, Professor Zishan Ahmad and Professor Soil Arshad. Thank you as always. Thank you, my Sinkwara Pharmaceutical Society of Nigeria, PSN, Parts and College Chairman, ESCO, fellows and family members. I do not take for granted the friendliness of the great Alpians and Malpians. It really means a lot to me that I found Ilorin costly despite my initial apprehension. Eshe Modupe to the elders, Agbanya Tubot Dale and me. Thank you, Auntie Memuna Zakaria Alukwade and her children for hosting me during my first months in Ilorin. To Alaja and Alaja Latifat, Yusuf Idolat, their sister and noble daughter Samina and Chope. Thank you to my neighbor, Dr. Oshunio and family. To my darling sister, Sajia Taibat Ali, Afusat, Bonatito, Sophia, Sulia, Sangania, you're all appreciated. And to my sister, Mrs. Raim Olani Olari, thank you for all you do. Thank you to my friends and neighbors, all here listed, right from Alaja Abudu, everyone listed here. It's a long list, but you are all appreciated, down to the kiddas. And thank you to the entire members of Women for Islamic Culture and Awareness. To my darling sisters and families in Leicester and Oxford, who hosted me throughout my PhD, the Bio Shoguns, Fatima Blazizis, Alice Baloguns, Kadaras, I know everyone is online, Afinoris, Unites, and the Muslim Women's Circle, the big Muslims of Motesh Shogun Kes. Thank you so much. My teachers and friends in the Arabic classes, Arabic tutors, Miratul Arabic, Konatil, and Rodatul Ilorin Academy. Thank you for making the language fun to learn. I am sincerely grateful to my Saudi Arabia crew, Dr. Mohabat Raji, Ridwan and Muminas, Malak and Rida Tijani, Alaji Alaraja Busari Ayola, Professor Baba Kamal and family, Haji Arafiat, Adeola, delegates and the Noibis, our Faisal University team, my Dean, Dr. Manal Ali, my sub-dean, Professor Mohammed Kamfal, and up to Ranim Abushema. Thank you so much. I thank my darling sisters, Lola Buki, Shola, everyone is here, Lola, Bisola, Islamia, Karila, Hawa, Mr. Medina Bioshogun, and everyone here. I thank you so much. And of course, my priceless 1992 set bodies at FGGC Owerri and their families. Fond memories of my friends who have preceded us: Kemi, Monsurat, Bushra, Aminat, Hamida, Sister Mimuna, Latifa, and Ganiya. May Allah continue to have mercy on your souls and unite us in Jannah. Amen. My big sisters, brothers, and their families, Auntie Bisola, Auntie Yemi, Auntie Fatima, Auntie Sharifa, Auntie Noimat, Auntie Jumai, Auntie Aisha, and others, and Dr. Wale Babalaki, Justice Wale Abiru, Alaji Tawid Amuson, Alaji Ali Uluwafui, Alaji Emefulawa, Shola Young, Alaji Kayode Olatunji, and Professor Yusuf Ali. Thank you much for always looking out for me. Thank you, sir and ma, Dr. Jubil and Alaja Fatima Uyekon for support over the years. I thank Alaji Latif Balogun for looking out for me as his daughter. Alaja Toibat Olatunji, thank you for the prayers I always look forward to hearing on the phone. Thank you to my sisters, to friends of my parents, family, hobby, and children for their love, support, and prayer always. Thank you to our support system, Tajuddin Agiri, Toka Adeleke, Kende Ajayi, Lydia David, Abdulrazak Mansour, and Achibong Edet for being dutiful always. I appreciate your wise counsel, Sheikh Abdulrahman Ahmed, the chief imam and his wife, and Ustaz Abdul Fattah Abdul Majid, Sadia Model School teachers, parents and management. Thank you for the foundation lady, my children. Thank you, Ms. Anio Loko of Minders Montessori and Tomache School for molding my children. My first friends, classmates and senior colleagues, now alumni group of Adini Jones Primary School, FGGC Owerri, 1992 set, Sisterhood Rocks, Lagos, Southwest and Global chapters of FGGC Owerri, Old Girls Association, College of Medicine, University of Lagos, 1997 and 1999 set, College of Medicine, University of Lagos, Unilag, and the Unilag Muslim alumni, the YBC No Limits 2004-2005 says, mentors and mentees at the Muslim Mentors Connect Group, members of NAPA, CPAN, MPAN, EMAN, Science Association of Nigeria Council members, Nigerian Society of Pharmacological Sciences and Experimental Therapeutics Respect, West African Society of Pharmacology, WAS, and Surveyors Wife Association of Nigeria. Thank you. Thank you to the board members, management, staff and parents of Christian Pulse Leadership Academy, CPLA, Abuja. Oh Allah, you are the most merciful. Shower your rahmah on the soul of my late husband, Ayodele, who encouraged me to engage in research in tune with the world development. You were indeed a source of encouragement. Your legacy lives on through the wishes and dreams you supported. You were the one who paved the path I'm treading today. Oh Allah, forgive his shortcomings and count him among the dwellers of Jannah. Amen. To my sisters, Sakina and Adama, whom marriage and Islam made us one, I appreciate you a great deal. To the children, Hulaima, Yasira, Amira, and Khalifa, you are greatly valued. May Allah continue to expand our rooms and grant us immense blessings. To my children, Hikmah, Mukhtar, Iman, and Alamin, and thank you for the joy and purpose you bring to my life. Thank you for being patient, resilient, and always understanding. May Allah continue to make you the goodness of our eyes. Ameen. To my crown, my sweetie, my Quran, my heartbreak, my wonderful and loving husband, so we will call it Kasi. You are the pillar of support that holds me firm throughout this continuing journey. Thank you for giving me the opportunity to keep soaring higher. Thank you for supporting and watching me bloom with every wing. May your days be long upon righteousness. Amen. Thank you all for attending physically and everyone watching virtually. You are most appreciated. I'm greatly honored as Sheikh people. Jazakumullah khairan. Alhamdulillah Rabbil Alameen.
Congratulations.
On behalf of all of us, I congratulate the 285th inaugural lecturer, Professor Adeola Tawakalitu Kola-Mustapha, FSAN, who has treated us unrooted in nature, refined by science, a new horizon advanced drug delivery. And on our behalf, I want to appreciate all of us here this evening. I start with our university scholars. Thank you so very much for coming. We appreciate the members of the governing council, Professor Abdulrashid Abiodun Adeoye, we thank you. Professor Muftau Adeni Ijaya, thank you for coming. Former member of council, Dr. Rashid Adeke Otola, thank you for coming. We acknowledge the presence of Professor Nasmat Surajuddin Bakinde, Vice Chancellor, University of Applied Science, Engineering and Technology, Gambia, who is with us online. We thank Professor A.W. Olatiwo, former CMD, UITH. Professor Noha Yusuf, Vice Chancellor, Alikma University, who is represented here today. We appreciate all the government functionaries that are here with us this evening. We recognize and appreciate Professor Yusuf Olawale, SCN, First Korean Gate of the Emirate. Thank you for coming, sir. We recognize Imam Abdurrahman Hamad and Haji Arabia Hamad. Thank you so very much for coming. The Chief Imam of University of Ilorin, Professor Nasir Abdusalam. We recognize the various professional and social associations. The list is really very long. All of you are well recognized. Pharmaceutical Society of Nigeria, Fellows of Pharmaceuticals, PSM Board of Fellows, etc., etc. We recognize Professor Philip Olawanmi Olurinola, the pioneer dean of Faculty of Pharmaceutical Sciences. I recognize the presence of my amiable wife, Ajia Amibat Adeola Egbewole. I welcome the wife of our mentor and Oga, Ajia Taibat Oba Abdurrahim. Thank you so very much for coming, ma. And also the wife of our former Oga, former Vice Chancellor, Professor Taiwo Ambali. Thank you for coming, ma. And I have a very long list of those to be recognized. I recognize you. Allah has also recognized each and every one of you. Let me especially recognize my in-laws, Alhaji and Alhaja Kasali. Thank you for coming. We have 264 attendees online. Thank you so very much for attending. I recognize respectfully the parents of the inaugural lecturer, Alhaji Adekunle Lasisi. Thank you, sir. I welcome Mama Alhaja Adeola Kasim. Thank you for coming, ma. I also recognize Sobio Femi Kasim, the father-in-law. Thank you, sir. Children of inaugural lecturer Ikma, Mukhtar, Iman, and Alami. Thank you so very much for supporting our inaugural lecturer. And of course, the person that has been described in flowery language, Sobio Olukolade Kasim, the spouse of the inaugural lecturer. Thank you so very much for coming. We thank each and every one of us that has made it to our 285th inaugural lecture this evening. God bless you all.
Shall we arise for the university anthem? Well, once again, we want to thank you all for coming. Thank you for being a part of the 285th and the series of inaugural lectures of this university. The third from the